Yeah, we are live. I over to Shabana madam.
A very good morning to one and all present here with one. I, Sayyida Shabana Sultana, Assistant Professor, Department of Pharmaceutics from RBVRR Women's College of Pharmacy, feel it is pleasure to be a part of this seminar. Before we move into session, let me first acknowledge the presence of our third Professor Sumakant Garu, Honorable Correspondent Professor K. Mutyamurthy Garu, eminent speaker, and all the registered delegates on behalf of RBVRR Women's College of Pharmacy to the webinar on artificial intelligence in drug discovery and healthcare system. Gatherings are not possible due to this pandemic, but COVID spread did not stop us from spreading the knowledge. Let me brief what this webinar is about. Artificial intelligence means the program of human cognition. Our goal of conducting this webinar is how artificial intelligence plays a role in solving the problems and improving the logical thinking in drug discovery and healthcare systems. It is mark of our underlying tradition of the Almighty at beginning of an important event. Let's revive the tradition by starting this session with a prayer song. Indeed, we all feel spiritually enlightened. Dear God, thank you for all the blessings you have bestowed upon us. We are truly grateful for them. Thank you for allowing us today to meet and share our knowledge and time with one another in this webinar. May you extend your divine wisdom to our speakers so that they would be able to impart effectively their God-given knowledge to all of us. May they be blessed as they continue to bring their expertise to people who needs them. Bless the participants as well so that they would be able to gain the vital information from this webinar. Now I would like to invite our principal and convener of this webinar, Professor M. Sumakant Garu, to give a welcome note. I welcome you, ma'am.
Yeah. Thank you, Shabana. Good morning to all. It gives me immense pleasure to invite all the speakers of this webinar, and today's speaker, Dr. Dev Priya Kumar, and welcome all the registered participants for this webinar for a week. I am glad to introduce the college, which is RBVRR Women's College of Pharmacy. It was established in 2006 by Hyderabad Mahila Vidya Sangam, a non-profit society. It was established with a vision of the society as to empower the girl students. The college was started with an intake of 60 students in B Pharmacy in 2006. Later, it got upgraded with courses M Pharmacy as well as PharmD. The M Pharmacy course was started in 2010, Pharma Chemistry as specialization. Subsequently, it was updated with other specializations like pharmaceutics, pharma analysis, and recently for this academic year, we got the approval for pharmacology also. And in 2017, we started PharmD 6 years course, and Continental Hospital is the hospital which is for PharmD course. we got the MOU with that hospital for 10 years. The college is affiliated to Osmani University and it is recognized as one of the research center under Osmani University. And the college got NBA accreditation for B Pharmacy course for 3 years. And it also got the certificate of IIC Institute Innovation Cell. And more than 100 publications are accounted for the college. The college is having well-nourished library with more than 8,000 volumes as well as the digital library. The college is having well-qualified faculty. More than 10 faculties having the PhD and the other 5 members got registered for PhD. With this brief introduction, we can say that within short span of time, that is within 14 years, exactly by this month, it is going to be completed 14 years. With short span of time, the college got such a reputation as well as upgraded with all the courses. Now it is full-fledged pharmacy college. The whole thing is because of the effort by the faculty as well as the cooperation with the society. Now, I would like to express my gratitude towards all the speakers who accepted readily to deliver the lectures for this webinar and all the participants who got registered for this on the trust of the college. And I welcome all the participants to attend for this webinar for this week. Now, coming to this topic which we selected for this webinar, that is artificial intelligence in drug discovery and the healthcare system. There are the two reasons why we have chosen this topic. One is to aware the pharma fraternity to have the scope for their career in artificial intelligence. The participant will come to know after these lectures or the talks what and where you can improve your career opportunities in this artificial intelligence. It is the opening for pharma field. Till now, we know that the jobs only in that field, which is nothing but either the pharmacist or in the industry. Only these things we know that. So other than this, now we come to know that what positions we can gain in our career will come to know through these talks. And the second one is to make aware of the scientists who are working in R&D. They should know that what technology is available in the market, how and when they can use these tools to reduce their research time as well as the cost of the research. So these two are the aspects make us to choose this topic for this webinar. Now coming to this artificial intelligence. So artificial intelligence is not a new term. Almost all the individuals know that. Why? Because in their daily life they are experiencing this artificial intelligence. So artificial intelligence is nothing but mimicking the human brain functions, nothing else. So in our daily life, we are experiencing since wake up till bed, all the things more than, I think, more than dozen we can experience through this artificial intelligence. So this only we know that. Now we would like to aware of what is the application of this artificial intelligence in drug discovery as well as the healthcare system. There are the devices or the modules of this artificial intelligence like machine learning, deep learning, neural networks, and natural language processing. So these are the tools which are mainly used for these two purposes with my knowledge, if I am not wrong. So not only the pharmacy field, we can also enter the other fields also. This reflects the things through these talks. So now what is the machine learning? Machine learning is nothing but the computer-based technology. It is playing a major role in drug discovery in multiple steps. So what are these multiple steps? It can predict the target structure. It can predict the biological activity, and it can predict the hits, the probability of the hits during the discovery. And it also gives the information about ADMET properties of the new ligands or the compounds. Now, recently, it is also used to improve the score functions in docking studies. So these are the major applications of this machine learning in drug discovery. Now coming to this healthcare system, so because of the availability of analytics as well as the statistics, a large data is available in healthcare system. So that itself is enough for this artificial intelligence to develop more models to make it easy for nurses as well as for doctors also in diagnosis or in assisting the patients. Now, the major areas where this artificial intelligence is nowadays is using neurology, cardiology, as well as the cancer. So these three major areas where they are using this artificial intelligence to make it simpler way. Now, the other modules like machine learning, the other things which are nothing but deep learning or the natural language processing and the neural networks. So these are mainly used in healthcare system to predict what is the type of the disease with that input from the data you can analyze. What is the probability of the treatment and what is exactly you can have it. That means it is the supporting system to the physician to diagnose or to prescribe the prescription. Now there are 5 major areas. The developments of this artificial intelligence in healthcare system. This is it assisted intelligence assisted robotic surgery, virtual nursing assistants, aid in clinical judgment, and workflow and administrative tasks, and image analysis. So these 5 areas are the major things of this artificial intelligence application. Now, so based on the information which we are going to get from the speakers during this week, we come to know that means what are the outputs? The outputs are nothing but what are the different procedures, how, where and when we can use in drug discovery as well as in healthcare system. Now, I am expecting the students, you can aware of your opportunities to settle your career in your life. So with these talks, I think at the end we can get the conclusion that in pharma curriculum, whatever we have at present, that you have to replace by introducing some softwares or the algorithms or the subject itself. I think it is better to introduce as one of the subject of this data science or the data analytics. So this is from my personal view. I can suggest that changes in the curriculum. And now you can get these machine learning programs online also. So our participants can utilize this time to learn something of this, and you can apply for this data analytics to get the job. Like computer science. So it is nothing but wherever the opportunities are there, not only for engineering students. So this, I think, the more appropriate person is the pharmacy person. Why? Because whatever the inputs they need to develop these machine learning models or the other models which are used in healthcare system. So the basic thing is nothing but the input. So that input is related to drug discovery or the healthcare system is nothing but only this pharmacy professional are the appropriate persons to help to the developers. So instead of having that complete engineering background student, if you have the collaboration with the pharmacy, I think you can save the time in developing these machine learning models. So with this, I can conclude myself. And once again, I welcome all the speakers for this webinar and today's speaker, Dr. Deva Priyakar. Thank you.
Thank you, ma'am.
Thank you.
Thank you, ma'am, for enlightening us with your words of wisdom. Learning is a treasure that will follow its owner everywhere. I'm sure at the end of this session, you will be happy for the knowledge you gained. Now, I would like to request Dr. A. Krishna Shailaja, ma'am, Associate Professor and Head of the Department of Pharmaceutics, to continue this session. Happy learning. Over to you, Shailaja, ma'am.
A warm welcome to all participants. Thank you, Shabana, for introducing me. It's my pleasure and honor to introduce Dr. Deva Priya Kumar sir to all the participants. Sir is working as Associate Professor and Head of the Department in IIIT and Hyderabad. Coming to his academic profile, sir completed his BSc from University of Madras. He did his MSc from Pondicherry University in Chemical Sciences. Sir completed his MPhil in Computational Chemistry from Pondicherry University. Sir did his PhD under the eminent guidance of Dr. G. Narhar Shastri in Pondicherry University. Sir did his postdoctoral research in University of Maryland under Professor Alex Mackrell. Then he joined with the IIIT Hyderabad in 2008 as an assistant professor. He continued his journey and later promoted as associate professor in the year 2008. In 2012. Sir then became head of the department in 2013. Sir held several positions, he got several honors. Sir was acting as a visiting researcher in University of Auckland, Glasgow, Scotland. Sir worked as an invitation fellow in IMS, Japan. He is working as a visiting researcher in various several universities. Coming to his awards and recognitions, Sir has received visiting fellowship from INSA and Royal Society of Edinburgh in 2019. Sir received Distinguished Lecture Award from the Chemical Society of Japan in 2016. Sir received Invitation Fellowship from JSPS 2016. Sir received an Indian National Science Academy Young Scientist Medal in Chemical Sciences in the year 2011. Sir received Young Teacher Award from AICTE. Like that, Sir has received several awards and gold medals for his work. Coming to his funding projects, Sir received funding from various sponsoring agencies such as DST, DBT, INSA, AICTE. Some of the projects I would like to mention over here. He received funding for the investigation of atomistic mechanism of co-solvent effects on protein folding equilibrium from DST SERB. Sir has received funding for investigation of physicochemical basis of stocking interactions in nucleic acid architectures from INSA. Like that, Sir has received funding for various projects, almost more than 10 projects Sir has completed. He conducted several scientific meetings. He acted as a convener for a symposium on mission learning for science in the year 2019 in IIIT Hyderabad. Sir worked as a convener for Indo-German workshop on computing in chemistry, biology and medicine in 2017 IIIT Hyderabad. He acted as a co-convener for theoretical chemistry symposium which was held in University of Hyderabad in the year 2015. Sir acted as a convener for the 21-day winter school on modeling chemical and biological activity in the year 2014 IIIT Hyderabad. Sir acted as a convener for International Conference on Chemistry with Computers in the year 2014 at IIIT Hyderabad. Sir held various administrative responsibilities at IIIT Hyderabad. Currently, he is working as a head for Center for Computational Natural Sciences and Bioinformatics. He is a coordinator for sponsored research. Sir. He is a faculty in charge for high performance computing facility at IIIT Hyderabad. He is also acting as a chair in these lectures. With this introduction, I would like to invite Dr. Deva Premkumar sir to deliver his talk on mission learning in interactive design. Thank you, sir. I cordially invite you to deliver your talk.
Okay, so first of all, I would like to thank Dr. Krishna Salija for a very nice introduction, and also Professor Nakran for and her team for inviting me. Is the audio okay?
Sir, it's giving disturbance, sir. Our voice
Some
is
disturbance is there.
not clear. Some disturbance is coming.
Yeah.
Say it again, please.
Yeah. Now. Okay. No.
Now it's okay.
But the disturbance is there. I think maybe in the system.
I did not do anything up. Hello?
Yeah.
Hello,
Now
sir.
it's
Is it
clear.
clear now?
Yeah.
Okay. I mean, something else is coming from someone. Can everyone mute the mics, please?
Maybe. Yeah. Better to mute all.
Okay, I think it's better now, yeah? Okay, I think so. Okay, good. So thank you, Professor Sumakant, and for organizing this nice webinar series, and also Dr. Krishna Shailaja, who has been I think working to put together this particular program, and I also would like to thank her for the kind invitation. Okay, so today I have shared the screen, so I'm going to use my slides. Okay, good. So so basically today what I'm going to do is give an introduction of how machine learning can be used for drug design, and maybe a little bit about how machine learning is useful in healthcare. Okay, so that's basically the idea. So I am from IIIT Hyderabad, and I'm from the center called Center for Computational Natural Sciences and Bioinformatics. Okay, so let's move on. All right, so when we say healthcare, right, we look at healthcare in these three major paradigms. Okay, so one is the prevention of any possible illness, and then if there is some illness, to be able to accurately diagnose a particular that disease, and then to have a treatment protocol to manage that particular disease. Okay, and the area of healthcare, right, has undergone a revolution in some sense, and then the kind of treatments available now, or the kind of techniques that are available for diagnosing diseases, and then the awareness that the system has created in terms of maintaining preventive health and so on, it's unprecedented. It's actually we are all much more aware than even 10 to 20 years ago, right? And technology has contributed to this particular this particular evolution of how the healthcare has shaped up, okay, or how it has evolved in the recent past. Okay, so in principle, right, what we want to why AI, or why artificial intelligence, and why machine learning, right? So we want to increase the efficiency of the preventive healthcare system. Okay, and what do I mean by that is that there is a mechanism that already exists, right? And we want to improve the efficiency of that particular mechanism. Okay, so for example, we do know that if we can modify, or if we can change our behavioral ways. For example, if I go for a run every day, or I exercise regularly, I know that I can keep away from, for example, diabetes for some time, right? So can someone help me, right? Modify, or can someone help me to make lifestyle changes, for example, right? So can technology help, or can AI help, right? So medical imaging, for example, if there is a fracture, we get X-ray done, or for example, if in case of cancer, we get biopsy done, and then the histopathology images are looked into, right? So in that sense, the medical imaging has played a major role in terms of diagnosing diseases. And how can we make this faster? So how can we, once we, for example, if we take histopathology images from the slides, right, how can we quickly say whether there are cancerous cells or not? Okay, and then radiotherapy for cancer patients, for example, right? This is a very, very complicated and much more personalized kind of treatments that is given to patients, especially ones who are in not so initial stages. Okay, so can artificial intelligence, machine learning can help. There's a lot of work that is going on in these particular areas already, right? Okay, so and then the second level is, what can we do new? Can we come up with new novel strategies, right? So that, for example, can help me prevent diseases. For example, maintenance of smart health records, right? Every patient who visits a hospital, right, or every patient is associated with a smart health record, and that helps in foreseeing things, right? So that are normally not possible by human, right? Can we, for example, predict outbreaks? Now we are in a pandemic situation where there is an outbreak. Okay, so can we make predict these outbreaks, right? And then for example, can we use multiomics and then use machine learning to diagnose diseases in a way that we are not doing currently, right? So that the diagnosis can be done at very, very early stages. Okay, so and in terms of treatment, can AI help in personalized treatment? And then can AI help in clinical trials, for example, and so on. All right. Okay, so these are some of the areas where AI, or the artificial intelligence, machine learning, has been making a big contribution in the recent past. In the I would say within the last 5 to 10 years. Okay. All right. So what what do we mean by artificial intelligence applications in health? Okay, so in principle, you have a large number of data that is available at multiple levels. It could be a genomic level, or transcriptomic level, or it could be at the molecular level, or it could be a diagnostic level. For example, blood work, or it could be any kind of data that we can think of, right? If we can use these data and then apply in several areas of healthcare. Areas. Okay, so today, of course, we are going to talk mostly about drug design. Okay, all right. So for example, one of the major applications of artificial intelligence is in diagnosis. For example, you can take the CT scans of the lung and then predict lung cancer. And then you can take the electrocardiograms, right? And then if you have a time series data, so you can comment, or you can assess the cardiovascular risks of given patients. And for example, you can take the images of the skin and identify skin lesions, right, automatically by using computers. And then also you can use eye images and then identify retinopathy and so on. Okay, so these are some of the applications in diagnosis. Okay, So what is possible? There won't be there won't be a situation. There will be a situation quite soon where at least there will be prototypes. There are already, for example, we wear smart watches which are capable of recording like ECG, for example, right? Heartbeats, right? Continuously. Heartbeats is measured continuously, right, by very very simple devices. Can we use this data to say something about the health of a patient, right? So basically here what we are seeing is like a number of sensors that attach to someone's body, and then how the data from these sensors can be used to diagnose diseases or to monitor health, right? So how does it work? Basically, if you have diagnosis done, or the images like x-rays or CT scans or fMRI scans and so on, if you have those images, and then the temporal inputs, maybe for example data coming from these sensors continuously, and then multimodal, basically like the proteomics or the genomic or transcriptomics kind of data, for example, right? If you can combine all of these and then use what we call a machine learning or artificial intelligence, and then be able to do a diagnosis which is much more efficient and much more early in terms of the diagnosis, which is good. Okay, so with this very short introduction of how where all AI ML can make a difference in healthcare, what we want to do is get into how machine learning is used in drug design. That's basically what we want to talk about today, right? Okay, so this is how my rest of the talk is arranged. Okay, so we will have a very general introduction of why artificial intelligence or why machine learning for for example drug design. Okay, and then we will try to understand in the most basic way what we mean by machine learning. When people talk about machine learning, artificial intelligence, right, this has become like every day. If you look at a newspaper once in 2 days, you will find this phrase somewhere or the other, right? It has become a very common talking point, and then these are also buzzwords, right? So we'll try to understand in very simple ways what we mean by machine learning. And then what I'm going to do is, I don't think I will be able to cover all of them. I will give some quick examples or case studies where the machine learning is used in drug design. Okay, so that's basically what we are going to do. Okay, so the first general introduction. Okay, so this is a slightly busy slide. Okay, so maybe I will use a smaller or a simpler picture in the next slide. Okay, so some of you may have seen, especially if you are doing pharmacy, or if you know about what a drug discovery process is, some of you may have seen a slide which looks something like this. Okay, so this basically explains how the drug design cycle or the process of drug discovery works. So it starts with a disease, and then identification of a target for that particular disease, and then ends with a medicine. Okay, so that's basically the that's basically the drug discovery process, right? It's a process by which you start from a basic biology knowledge and then end up with a drug molecule that cures a certain particular disease. Okay, so let's look at the same thing, but a simpler version, right? So as I said, once you have a disease, for example, novel coronavirus 2, right? So you have a disease, right? Now what do I want? What I want is a drug molecule that can cure this disease or treat this disease or to manage this particular disease. Okay, so that's basically what we want, right? So for that, what we want to do is to do what we call a simple biology. We want to understand the basic biology of this particular disease, saying, okay, if I take this disease, how does it, what happens, what is the basis of this disease, what are the molecular pathways involved, and things like that. Okay, and then the second one, once this biology is understood, you identify what is called a target. What do you mean by a target? so that there is some particular protein molecule, for example, right, usually a protein molecule that is involved in the pathway in that particular disease. If you stop the protein from doing its work, right, for example, in coronavirus 2, if some of you have been reading RNA dependent RNA polymerase, right? That is a protein which is important for making copies of RNA from the virus. Okay, so if I'm able to stop that particular function, the virus may not be able to replicate, right? Exactly the same thing. So have a disease and identify a protein which, if I stop its function, I can manage that particular disease. So identify that particular protein molecule, right? And then you perform what is called a drug design, right? This could involve computational methods. It could involve experiments. It could involve in vitro, in vivo studies, and so on. And that's where you identify molecules, right? Drug-like molecules that may actually bind to this particular target. Okay, right. And then this and then the next stage is the clinical trials, right? Preclinical, and followed by that clinical trials, where you have a process by which one demonstrates that this particular drug molecule that you have come up with at this particular stage is actually safe. Well, that is one major point. And then the second point is that it actually works. If I say that this particular molecule is actually working against this particular disease, right? This is the process by which these two mainly are assessing that this molecule is actually safe, and then it actually works in terms of treating that particular disease. Okay, so once all these things work out, and then you have a marketable drug, right? So now the question is, what is the role of artificial intelligence here? It looks like we have a process which is very well designed, and it seemed to be working, right? One of the things that some of you may have heard, that this particular process is very long in terms of time, and then it also is very expensive, right? It costs a lot of money before you can get to this particular point, starting from here, right? So this is something that we all have heard of. But the question is the question that we want to answer today is, why do we need, or why do I need artificial intelligence in this particular context? Okay, so let's go to the next slide. Okay, so let's say, right, so I'm in this slide, I'm just going to talk about few things that are just from the artificial intelligence or the machine learning viewpoint. All right, so let's assume that we have a new disease, and then a basic biology is understood, let's say, right? What if there is an algorithm, right, which can take this particular data and comes up or identifies the target? Okay, so as you can see, I have something called a drug target identification using a machine learning approach. Okay, so what if once this is done, what if as an algorithm takes this particular target or takes this information and then comes up with high quality protein structures? Okay, so that's basically protein structure prediction from deep learning, right? So once I have that, right, is it possible for me to take this particular protein from here that was designed by artificial intelligence and then generate new molecules, right? Similarly, I can talk about new things. Once I have a molecule that is proposed by an algorithm, can you have an algorithm which can tell me how to make it in the lab, right? I want to at some point I have to make it and then test it, right? And then can I have some robots which will take the information from here and then actually do the synthesis for me? And then can I have some algorithm which will confirm or which will validate the synthetic process that is followed by this particular robot? And then can I automate testing of these molecules that I get from here for further analysis and so on, right? So all the way up to can machine learning help predict clinical trial outcomes, right? So in principle, in the last few years, especially last 2-3 years, we have been seeing that like this particular, the whole cycle has a lot of contribution coming from artificial intelligence in a big way. So if all the papers that I have quoted here or they referred here, all of them that have been published in the last within the last 2 years or so. Okay. All right, so this is in this context what we are trying to discuss, where again we talk about deep learning, machine learning, and artificial intelligence. That's basically the context. All right. Okay. So as Professor Sumakant was saying, right, we see AI ML applications everywhere now, right? Whether we are traveling or whether we are someone is doing a marketing or in social media or like Facebook, Twitter, and so on, like automation, like self-driving cars, and then the credit companies or banking, and then insurance companies, and then driving sales, right? And one of the major applications, as I said, is healthcare. All right. Okay. So as you can see in this slide, this basically is the number of startup companies in the area of artificial intelligence applied to drug design, right? From 2017 to 2020. As you can see, this number has increased, significantly within the span of 2.5 years. Okay, so that kind of gives us an idea about how the artificial intelligence is coming in a big way, contributing or starting to contribute in a big way to the drug discovery process. Okay, so now once I hope I have been able to give a some picture of what is the relevance of artificial intelligence and machine learning in drug design. Okay, all right. So now let's try to understand what we mean by machine learning in very, very simple ways. Okay, so what we are going to do now is this. So we are going to do a simple task now. Okay, so the task is this: an image is going to be shown, right? Let's assume somebody is going to show me an image, and that image has one of these three characters. Okay, 1, 2, or 3, right? The image basically has a character. It's a Chinese character, right? And then it will say either 1 or 2 or 3, right? So now we have to design an algorithm which will tell us whether it's 1, 2, or 3. That's basically the idea. All right, so now we are going to take two approaches. Let's take the first approach and then say we say some rules, saying I'm going to have some rules which using which we identify whether it's a 1, 2, or a 3, right? So the rule is that if there is one horizontal line, that's 1, right? So what does it mean? One horizontal line is like this, right? That's a horizontal line, right? If rule 2 is that if there are two horizontal lines and the top line is smaller than the bottom line, it is 2, right? So then says like you want two lines, one, and then there is a top one which is smaller than the bottom one, right? So that's 2. And then rule 3 says if there are 3 lines now and the middle line is the smallest, top one is medium, and bottom line is the longest, so basically I say okay, possibly this is 3, right? So this is basically what we have done now is we have given a set of rules, right? For the 3 characters, right? You have rule number 1 which says one horizontal line, rule number 2 it says something, and then rule number 3 it says something else, right? Based on those 3 set of rules, we have to identify what that character is, right? If based on this, if I go here and then if I ask you what this is, right, you will quickly say that's a 3, right? That's basically the rule we have explained just now. And then this is 1 and this is 2, right? It's pretty straightforward, right? And based on the rules, I'm able to actually say it, no problem there. So now I'm saying like, okay, I don't like that particular approach. I'm going to follow a different approach. Okay, so the approach is that I'm going to show these particular images, saying these are 5 10 different ways of writing number 1, right? All these things, right? These 10 different characters that are shown here, all of them correspond to number 1, right? So similarly, I show you saying, okay, all these 10 different characters correspond to 2, and all these 10 different characters correspond to 3, right? So now I have, instead of giving you rules, what I'm saying is I'm just giving you some examples, saying, okay, these are the examples by which maybe you can learn yourself, right? So if I look at it, my cognition, or my brain tells me that, okay, if there is a horizontal line, that means it's a 1, and then if the character looks like one of these, it is number 2. If it looks like one of these, then it's number 3, right? If I go back and do the same exercise and then ask what these 3 are, one will be able to exactly say that the first one is 1, second one is 3, and the third one is 2, right? So in principle, what we have done is taken 2 completely different approaches to understand what we mean by machine learning, right? Or to solve that particular problem of identifying the Chinese character, right? So in the first one we use certain rules, right? And in the second one, someone is actually learning themselves based on examples. That's analogous to the traditional computing, and this is analogous to machine learning. Here we exactly showed or gave rules in terms of what these characters are. So in this case, we did not give any rules. We just gave some examples, and based on those examples, we learn ourselves in terms of understanding what these things are. Okay. All right. Okay. So how do we understand? This is like a traditional computation. All right. So I have a software program, right? And I have a certain input, right? I give this to a computer, right? And the computer takes the program, executes it on the input, and then gives me an output, right? So that's usually what is done, right? So for example, if some of you has been some of you are familiar with for example, computer aided drug design, right? You may have heard of this software called Autodoc, or for example, Schrodinger, right? So you have this program, right, which is installed on a computer, right? And then you give input. What are the input? The 3D structures of the protein, and then the ligand, and then the computer will take this this information and uses the program, and then gives me an output. What is output? Basically the binding affinity or the scoring, docking score, right? And maybe the structure of the complex and so on, right? So in machine learning, what we do, as you can see here, what we have actually done is, I have taken this and then moved it on this side. I have taken this and moved it on this side. That's the only difference between the two, right? So here, so what I'm doing is, instead of giving the input and a computer program, what I'm saying is I'm giving the output, the past output, right? Or large number of these outputs and the corresponding inputs. And then I'm asking the computer, instead of saying what will be my output, what I'm asking is, you give me a program, or you give me a computer program that will take this as the input and will give this as the output. So that's basically what we are asking. Like, in other words, what we are saying is that I'm giving the computer a large number of binding affinity values, or the docking score values, right? And then I'm also giving the computer the protein ligand structures, and asking the computer, saying, okay, if I use these as the inputs, this is the output I should be getting, right? Can you take these two sets and then tell me what kind of program, or what program can connect these two, right? So that's basically the question we are asking, right? And now, and that is usually what is called a machine learning model, right? So once I have this model, right, I can take this, okay, I can take this and put it here, and then use it in a traditional computational sense in the future, right? Instead of this being a program based on a rule, this program now is based on the learning, the model, or the machine learned model will be my program. Okay. All right. So okay, so this is what in principle we are doing, right? So normally in sciences, right, whether it's for pharmaceutical sciences or in physical sciences, chemical sciences, biological sciences, it could be any sciences. So this is the fundamental paradigm in which we do work. We do experiments and then we make observations. Okay, and then based on these observations, we try to understand this particular phenomenon, why we observe certain thing, right? So that's basically a theory, right? We understand, and that's the theory we have. And then once the theory is solid enough, we use that to write programs and then do computations and then predict new things. Okay, and then this cycle continues, and then when this cycle is mature, then the experiment is actually able to contribute to real life applications in that particular area. And where is machine learning here? This seems to be again a solid cycle which is working for a really long time, right? So now the machine learning comes here. Okay, so instead of formulating a theory, one option is use the data from here and then create a machine learning model that can be used to do computations and then improve the experiments and try to see if we can improve the real life applications. Okay, so now back to machine learning applications in drug design. All right. Okay, so this is not very new, right? For example, if you see, this is a paper from 50 years ago, right? So what did this particular paper do? They basically took a set of these molecules called dioxanes, okay, 1,3-dioxanes, okay, and then they, with respect to different R1, R2, R3 groups, okay, so they assessed their pharmacological activity, right, based on a perceptron network, right? This sounds very, very similar to the modern machine learning methods, right? Okay, so that's basically exactly what they did. This is a machine learning approach for drug discovery, and that was done 50 years ago, right? So, and what changed in recently? Why we are making so much noise in terms of ML applications in drug design, or in, for that matter, any other fields? Now, that's that we have to discuss a lot more and to understand what is new, what is why there is so much noise, and why the applications have become more effective and more efficient. Okay. All right, so that I hope I was able to give a simple and understandable, simple and understandable definition of what machine learning is. Okay, so that's, of course, is at understanding at one level, right? We want we have to go into much deeper, and it is much more than what I'm trying to simplify. Okay. All right, so let's say I'm going to use the next 20 minutes or 25 minutes or so, and then let me quickly check my. It's up to 12:15. All right, so we have about half hour. Maybe we can discuss quite a bit in terms of how few more case studies. Okay, so the first case study that I'm going to very briefly discuss or mention is protein structure prediction. So this is some of the things are done were done in my lab, and all these things, but not this particular work. Okay, so I chose this example, I think, because I wanted to discuss at least one work that is done by some other group, and also that the one which made a lot of news. Okay. All right. So what is protein structure prediction? All right. So for that, we quickly, briefly try to understand what this picture means. Okay. So when a protein is synthesized, whether in a biological cell or in a laboratory, right? When the protein is synthesized, right, and if the protein or if the sequence corresponds to a globular or in a functional protein, or or a protein which has a definite structure, so they undergo folding and then forms a native structure or a unique structure given that particular sequence of the amino acids, right? So what are amino acids? They are in principle a large what is this long line? So that's basically the representation of a large number of amino acids, okay, so which are connected by peptide bonds, right? It says linear long polymer, right? So now this undergoes folding, so that means it folds onto itself by forming different intramolecular interactions, like the hydrogen bonding, ion pair interactions, hydrophobic interactions, and so on, right? And then based on that, they form a given definite structure, right? This process is reasonably fast, right? Once the protein is synthesized, in most cases within 1 second, a native structure is formed, right? So why is this important? When if you remember a few slides ago, I talked about target identification and then find a drug molecule for that particular target. Okay, so usually proteins are the target molecules. Okay, but getting the structure of the protein is not so difficult. That's not so easy, right? And hence it is desirable to have theoretical methods or computational methods that can make this particular process predictable, right? So in other words, if someone gives the amino acid sequence, can you tell me exactly what the structure would be, right? So this is okay, so this is a movie which will give a little bit more information, because so basically this is a long peptide. Okay, so long peptide, and that's the protein, or that's the free energy surface, right? And then as you can see, it's nicely folding. The structure changes. I'll go back again. Right, so that's the the energy, or let's say the free energy of the peptide in this particular state, and then the movement of that black dot, right, tells the free energy change with respect to change in the structure. Of course, this is hypothetical. Okay, right, so that's the free energy. It's changing nicely. It goes down and then it starts forming a structure, and at the end it forms a nice three-dimensional structure. That's basically the process of folding, right? So there was a paper, some of you may know that there is this competition called CASP that happens. It's a conference and competition which happens once in 2 years, where a sequence is given and then researchers are asked to predict three-dimensional structures of these proteins. Okay, only based on the sequence. No other information is given. Okay, so what we have here, right, is the structure of 3 different proteins. Okay, and the ground truth, or the actual three-dimensional structure obtained from experiments, is given in green, and what was predicted by an algorithm is given in blue. As you can see on all the 3 cases, that the differences between the 2 is not much, right? So how did they predict? Okay, in addition to using informations like the evolutionary information of the protein and like the Ramachandran plot and things like that. So they use machine learning in a big way to make this particular prediction, right? So this particular group, right, you can see more details in this particular paper. Okay, so this particular group won the CASP competition by predicting 25 out of 43 structures reasonably well, right? And then if you look at the runner-up in this competition, they predicted 3 out of 43. The gap is too big, right? It's a wide gap between the winner and the runner-up. Okay, so it turns out that one of the reasons, there may be other reasons, but one of the reasons why this particular method was more successful than the others is because the way the machine learning was actually used. It's not that the others were not using, it's just that the more efficient way of using this particular algorithms. Okay, so again, so this is from that particular group, right? So how they were able to use, for example, a simple gradient descent method as part of after the the convolutional neural networks and so on, and how they were able to predict the three-dimensional structures of proteins very well. Okay, so that's that's a quick first case study that I wanted to give, right? Here, this is much more relevant to direct drug design, right? So first is molecular property prediction. What is molecular property prediction? Given a molecule, right? So given a drug molecule or given a small molecule, we want to predict the properties very well. For example, if you give me a molecule, maybe I want to know what exactly the log p value for that particular molecule is, and possibly I want to know what is the affinity of that particular molecule to a given protein, for example, right? We always are in the business of predicting molecular properties, especially in drug design, because we conceive a large number of molecules and we keep predicting or calculating their properties. Okay, so that's the second thing that I want to talk about, and I'm going to give possibly give two examples, right? So one is reasonably straightforward one, or a simpler one to start with. Okay, so this background may not be necessary for this audience, so I'm gonna skip to this. Okay, so in principle, right, if I want to do any property prediction, right, in the traditional sense, we use quantum mechanical calculations or molecular dynamic simulations, free energy calculations, and so on, right? So these are some of the most popular methods by which we predict properties of molecules, right? So quantum mechanics, right? Quantum mechanics is one of the methods which is considered to be like very, very accurate, right? This is one of the methods which is considered to be very, very accurate. Okay, so now the problem with quantum mechanics is that the the calculations are so complex that I have to spend a lot of computational time just for calculating some properties. For example, let's take energies. Okay, so that's going to be very, very difficult. So what do? At some point, with respect to the number of molecules, we say that I cannot do quantum mechanics anymore, right? And then hence I will start doing molecular mechanics, right? That's what we normally do, and that's what is acceptable, right? But then the question is it possible to come up with something called the machine learning method, right? Which basically gives very good answers as equivalent to quantum mechanics, but then in terms of the complexity or in terms of the time taken by these calculations will be as small as molecular mechanics. Okay, so that's basically what would be nice to have, right? So I want a method which is very, very accurate, but at the same time it takes less time. That's basically what I want, right? So that I can apply it on large molecules, and at the same time I can achieve high accuracy. All right, so what it is, this is work from my lab, right? So this is what we did. So first for a machine learning problem, right? Okay, so one thing I did was to skip a slide, which to think of it, I should have not done. Here. Okay. So in the machine learning framework, okay, so this is data driven, right? So if you remember in the first task that I gave, right, what I said was that I show you some examples of these images. Based on those images, you learn yourself and then try to answer the question, right? So that's the thing we did. No, so that is using those data. That is what is called data, right? That's an example of a data, right? Use that data to train a model to learn itself, right? That's what I mentioned as self-learning, right? So I train a model to train something, right? And then also to test whether that actual model works or not, you have to keep something for the test, right? So in principle, what we are trying to do is, in the machine learning, typically in a machine learning exercise, what we are trying to do is to come up with this particular function, which if it operates on the input, you will get the corresponding output. What is the input? In our case, it could be a molecular representation, or how the molecule is represented. Okay, so let's take a very simple example, right? So let's say these are the values of x, these are the values of corresponding y, right? So let's say I tell you, for a value of 1 for x, the value of y is actually 5, and then I say for a value of 20, this value is 99. If I say this value of 15, this is 76. For a value of let's say like 30, this value is 150, and so on, right? So if someone gives you this set of values, x comma y, right, and then if someone asks you saying, okay, so I've given you these examples, right? So now I'm asking you for a value of 10, what would be y, right? So what we will normally do, we will try to see what is the relationship between these two, right? If I look at 1 and 5, 5 is 1 plus 4, it is also 5 times 1. If I go here, 20 plus 4 doesn't work out to be 99, but if I say 20 times 5, it's actually 100, but it's 99, very close, right? If I take 15 times 5 is 75, 76 is very close. If I take 30 times 5 is 150. So in all these cases, right, if I take y and then multiply it by 5, okay, I seem to get what y is, right? So now, if someone asks you what this number is, most probably you would say this number is 50, right? So what is it that we have done? Based on these examples, I've tried to figure out what is the relationship between x and y, right? And then based on that relationship, I'm trying to answer this particular question. That's basically what we have here. So based on given values of x and y, right, I'm trying to figure out what is the relationship, or what is the function that will take me from x to y, right? That's basically the machine learning framework, right? Let's go back to that particular slide now. Okay, so now what is x here? X is the structure of the molecule, right? What is y, right? Y is the molecular property. Given x, or given the molecule, what is the molecular property, or what is y? Given x, what is y, right? So we are trying to discuss what x is here, right? So I want to define a molecule, right, to the computer. How do I do it? So the one way that we came up with, and there are several ways of doing it. One way that we came up with is like this. For example, if I have this particular molecule, which is a formamide, right, every atom is represented by an 8-dimensional vector. Okay, so the first 4 dimension captures what the atom is. For example, if this is a nitrogen, if I have to define N4, that's a nitrogen. So that'd be 0, 0, 0, 1, that's nitrogen, right? and then this defines the atom type or the environment in which the atom is present. This basically means this particular nitrogen is connected to 2 hydrogens, 1 carbon, 0 oxygens, and 0 nitrogens. That's basically what this means. So this 8-dimensional vector, okay, so this 8-dimensional vector captures the atom. Okay, so now, I'm saying, okay, I know how to define an atom now, right? That's the 8 dimensions. I take that 8 dimensions of one atom and then the 8 dimensions for the second atom, and then define a bond like this. So the first atom, second atom, and the distance between the 2 atoms. Okay, so this is a 17-dimensional representation for a bond. So similarly, if I take an angle, it is formed by 3 atoms. Like I take atom 1, atom 2, atom 3, and then the angle between the 2 bonds. And then I can have non-bonds, like non-bonded interaction, long range interactions, or the interaction between atoms that are not connected directly. And then dihedrals, there are 4 atoms followed by the dihedral, and so on. Okay, so that these are my x values now, right? I have to take these x's now and then predict molecular property, or y, which is the molecular energy. So I start from the structure of the molecule in the conventional sense, where you have the Cartesian coordinates, and the atomic numbers of each of those atoms in the molecules are given. Okay, and then I make a list of bonds, angles, dihedrals, and non-bonds, and then I use a deep neural network to predict the energy of the molecule, right? So that's basically the idea. Okay, so I use neural networks, okay, by taking the inputs. I for known outputs, that is E here, okay, I am trying to train this network so that it predicts or it takes these as the inputs and predicts the output. All right, okay, so that's basically what we do. And then I'm going to show quick examples, maybe this one, right? This is the energies of a set of some random molecules, which with a molecular formula of C11H22. Okay, so this is what is obtained by using a density functional theory calculation. It's a very high level theory calculations, which is often considered to be a good level of theory for predicting molecular properties. We normally do not use this in drug design because the number of molecules we treat during the drug design exercises is usually a large number, and hence we do not use this because of the computational time, right? So this is the method that I tried to show, which is the machine learning method that we have come up with, right? And if you see, right, the difference between the actual value or the value obtained from DFT and this one is very, very small, right? If I for a baseline, if I consider a semi-empirical method, which is considered to be a better method than molecular mechanics method, right? If you look at that one, that is not so good, right? So basically the idea is that I'm able to use machine learning to predict properties of molecules which are much better than even intermediate methods, right? Like semi-empirical methods, right? Forget about molecular mechanics. All right, maybe I'll show you one more example, right? So here we have taken very simple organic chemistry reactions like intramolecular hydrogen bond transfer, and then hydrogenation reaction, Diels-Alder reaction, aldol condensation, esterification, and rearrangement reactions. Okay, if I look at the reaction energies, calculated using high-level theory, right? They are like this. Okay, these are the 5 different or 6 different reactions, right? And if I take the machine learning method, you can see that they're almost the same, right? Except the difference here is the largest, and that reason we understand why it's the largest. We will not go into the details, but if I forget that, you see that the numbers are reasonably good, right? And if I look at this particular method, you see that they don't do that well as compared to the machine learning method. Okay, so that's the second case study I wanted to show, that you take molecular structure and use the molecular structure and predict molecular energies. All right, so the second example I want to take is to predict calculation of solvation free energies. Okay, solvation free energies, or I don't know if I have. Okay, no. All right, so solvation free energies are basically that when you have, let's say, a drug molecule. Okay, so let's assume that I have a drug molecule, right, in the gas phase, like it's gas phase drug molecule, and that goes to the aqueous phase, and that change in the free energy, right, that's the solvation free energy. So basically, the if this is large negative, that means the drug molecule is very soluble in water, right, and if it is not so soluble, right. Okay, this is the hydration free energy. All right, so the one which I have showed, this is aqueous, so it's a hydration free energy. It could be any other solvent, it could be hexane, it could be DMSO, it could be anything, right. So the change in the free energy when you go from the gas phase to the solution phase. All right, so why is that important? That is related to the ability to be able to or the property or the solution free energy of drug molecules is dependent, it strongly influences the absorption and distribution profiles of the molecule, right. So as you know very well, in addition to whether a drug binds to a target very strongly or not, right, so the pharmacokinetic properties, that the ADME and toxicity properties are also equally important, right. So and hence this becomes a important exercise in drug design most of the times. For example, prediction of log P and things like that, right. So usually if we want to calculate exact solution free energies, this is very, very difficult. The typical methods that are used are thermodynamic integration and free energy perturbation methods, which are molecular dynamics methods. Molecular dynamic simulation methods takes a lot of time, right, and complex, and also their accuracy depends on the force fields used and things like that, right. So here what we propose is a method which is based in machine learning to predict very accurate solvation free energies. Okay, and we also make it chemically interpretable. But yeah, okay, so this one looks a slightly bit more complicated. We'll not go into the details, right. So the idea is as similar to before we had molecule to energy. So now solute and solvent to the free energy of free energy change. Okay, that's basically what we're doing. So we have a solute molecule, let's say a drug molecule, and then a solvent. It could be water, it could be octanol, it could be anything, right. With respect to these two solute solvent combination pair, what is the solvation free energy? That's basically what we want to do here, right. So we use what is called a message passing neural network, graph neural network. So we have three phases here. One is the message passing phase, and then the interaction phase, and then the prediction phase, right. And we will not go into the details, and the results are here, where this plots the predicted solvation free energies versus the experimental solvation free energies. As you can see, the prediction is very, very good, and if you look at the root mean square error, that between the predicted and the experimental one, it's 0.57 kcal per mole. That's really, really good. Okay, right. So and then as I also said, this model is chemically interpretable. For example, if I take 2-aminoethanol as a solute, right, and ethanol as a solvent, okay, so I calculate something called an interaction map. Okay, so that is basically this one right here. Okay, if I take that interaction map, okay, and then look at the numbers, right, and then those numbers turns out to be chemically interpretable. For example, this number is the highest, right, so that corresponds to the interaction between the oxygen of the solute and oxygen of the solvent, right. Of course, this has a hydrogen attached to it, this has a hydrogen attached to it, and that would correspond to a hydrogen bonding kind of interactions, right, which is favorable. That's why it's very high, right. If I take N and O, similarly here, this is a polar group, this is a polar group, they can interact favorably, and hence it has a large number. Okay. But if you look at the other interactions, which as best would be called hydrophobic, those contributions are small, and hence this becomes chemically interpretable. Okay. So I will not go into further details of this, but the idea is this, that by taking the solute, right, that is a drug molecule, and then solvent, it could be water, it could be octanol, or it could be any other organic solvent. For this particular combination of solute and solvent, you predict what the solvation free energy is. Okay. All right. So now we have maybe 5 more minutes. Let's see. So the third thing or the fourth thing that I wanted to talk about is what is called inverse molecular design. So what do you mean by, okay, I'm gonna skip this particular part and go here directly. So what do you mean by inverse molecular design? So usually what we do is ask this kind of question when we do an experiment or when we do a computation. This is what we do. We take a molecule, right? We take a molecule, and then we do a computation, or we may also do an experiment. Okay, we may also do an experiment and then say, and then ask, what is the property of this particular molecule, right? But that's not what we always want, is it? Right? So because we usually want, what we want is this. We want, usually the question that we ask is that I want a molecule with a certain molecular property. So what that means is you already know the objective, right? Right. For example, if I have a RNA dependent or RNA polymerase of coronavirus 2, what the question that I ask is, I want a molecule which will bind strongly to this particular enzyme or protein. Right? That's the question I ask, not the other way around, right? So the how do we go about doing this? Okay. So there is one particular method called reinforcement learning. There are also other methods like the variational autoencoders and something called GANs, that can be used for these purposes. But what I'm showing today is the use of something called a reinforcement learning for molecular generation. Or in other words, instead of asking for a given molecule what is the property, we are going to ask, instead, I want a molecule with a certain property. The property is fixed. What is changing is the molecule. Okay, so we ask this software or the algorithm to suggest molecules with a certain property. That's basically what we want. Right? We just take a simple scenario. Okay, let's say I go to I get a new dog. Let's say, right? I have I wanted a pet, right? I wanted a dog, right? So now I want to train the dog. Okay, so let's say this is me here, right? Oh, what did I do? Okay, yeah, this is me here, right? So now I want to train the dog, right? So for example, I want the dog. Let's say the dog is standing. Then I want the dog to sit. Okay, I instruct the dog saying sit, right? Of course, the dog possibly do not understand in the initial stages, right? So it doesn't understand what I mean, right? But it does understand that I am commanding, or I am asking the dog to do something. Okay, that is clear to the dog, right? So let's assume the dog was standing, right? And the dog decides to do something, or it does an action, right? Maybe it moves, or maybe it lies down, or it turns around. It could do anything, or it can sit also, right? It does, from an initial state, right? The dog changes the action with respect to the command and gives a new action, right? So I, as what do I see? I only said the dog to sit, right? I see what the dog has done. If the dog did not sit, I'm not happy, right? Let's assume the dog actually sat and I'm happy. Then what I would do, I would possibly give the dog some kind of treat, or I would reward the dog. Okay, so what I mean, I would reward the dog or give the dog a small treat. Okay, so that's basically what we are trying to do here. So let's say I have the generator, a model or a machine learning model that is capable of suggesting new molecules. Okay, so it is possible to train, right? And then it can actually suggest valid molecules. Okay, so now I have a predictor. And then the predictor takes that molecule from the generator and then predicts what the property of the molecule is. And then if the property is close to what is being desired, then it gives a reward to that particular model. Okay, so in that way, this particular model will learn what is being asked. Okay, so that's basically some rough idea of how this algorithm works. Okay, so I will just quickly show an example and then we should stop. Okay, so let's say I have two properties. Okay, so this is first one is called log p. Some of you may be aware of. Okay, so log p is a quantity which helps us examine if a drug molecule can enter a cell or not. Okay, if the value is between 0 to 5 or 0 to 4, that means it is possible for the drug molecule to go from from outside the cell to the cytoplasm or inside the cell, right? And then that's one property, right? And then the second property, let's say, is we can as computational people, we can suggest a large number of molecules. It's easy to suggest, but someone has to actually make the molecule in the lab for it to be tested, right? So there is this method, or there is this score, right, called synthetic accessibility score, right, which which can take a value of 1 to 10. If the value is close to 10, it's almost impossible to synthesize that particular molecule. And if the value is lower, that means it's reasonably easy to make that particular molecule. That's the second property, right? So now I am asking the machine learning model saying, okay, so I have decided that I want molecules with a certain value of log p, and then I want the same molecules to have a lower value of this particular score, right? So if I go back and say, if I look at this, right, you see that if I normally generate without having to worry about any bias, right, this is how it looks like. As you can see, not all the molecules are in the range of 0 to 5. Okay, if you look at here, they are reasonably easy, but I will be happy if it comes in this region, let's say, right? Or the molecule can enter the cell only if they are in this window. If they are in this window or in this window, they cannot enter. Similarly, only if the molecules are towards the green smiley, then it's easy to make it, or else it will become very difficult to make, and hence this exercise is not so useful. So now I turn on this particular now I introduce the bias and say I want all the molecules to be in this range, and I want it to be as small as possible, right? As you can see now, the machine learning model has learned that it has to generate molecules in this range and then with low values here. Okay, so you can also extend this to many other properties, and then you can make it work based on the objectives that is being desired. Okay, so I want to quickly summarize. As I try to impress upon, modern machine learning methods seem to be fundamentally changing the way we do computations. And then I just gave a small number of examples, and then I try to give some impression on how this may affect. Right? As I said, AML methods, they do have a great potential to make the process much more efficient, like faster and less expensive possibly. Okay, so with that, so I would like to stop here and thank you for your attention. All right, thank you very much. If there are some questions, maybe we can take some.
Thank you very much, sir, for your excellent and wonderful presentation. We really enjoyed the talk. You explained the concepts of machine learning, how it will be useful in drug design. and the healthcare system also. You explained about various process and principles involved in the drug design, how AI is helpful and how machine learning is helpful. Some of the participants has raised a few questions, sir.
Yeah.
For that, I request Principal Madam, is there any queries from your side? You would like to say anything to Sir, Madam?
Yeah. One question, sir.
Yeah, please.
See, can you suggest for a pharmacy graduate to develop complete machine model which is useful for drug development? See, whatever I think as per your slides, the engineering background must be there. But without that, as a pharmacy graduate, by doing some courses, can he or she able to develop the machine learning model?
Yes, with the the modern paradigm of programming is to not actually write everything by yourself, right? So because with a very little anybody now who can spend little time on finding out what algorithms are there, what libraries are there, and so on. I think it's you don't to start with, at least, or to perform simple machine learning tasks, one need not have a very strong programming training, right? Of course it is essential, but it is actually possible for a pharmacist or students from pharmacy to be able to cope up with this, right? Because those days when we write a program, right, we write everything, all the operations, all functions we write explicitly. So nowadays, if you see, a large number of these libraries are available, so we just use them just like that. You just read them and then we use them. Especially in Python, you have large number of these libraries which we use, and then of course we also should be able to reuse these codes. For example, whatever we publish in machine learning so far, if you go to our web page, you will find all these codes are published, right? So if somebody wants to take this code and then reuse it to predict some other property, it is still possible, and to change that code to do something else, right? Reuse of the code is it's reasonably easy, right? So there's like, you can use like modules. You can take these modules, put them in different orders and things like that. It is possible. Of course it needs some effort, but it is possible.
Yeah, in point of view that where exactly you can accommodate this pharmacy graduates, undergraduate or graduate?
I mean, it's that the push has, there are a lot of programs which this online lectures and online videos, which there are a lot of them which can which is very, very useful, right? We also are, we also learn from resources like that, right? And then if we take simple papers where people have used machine learning for drug discovery, and then take them and look at that code, and then that could be a very good starting point, right? And of course that the good starting point is to take a paper and try to reproduce it. That would be the first point.
Okay.
Yeah.
Okay, sir. Thank you.
Sir, there is a question from Jessica.
Please tell. No. I think I can't hear. All right, I'm going to the YouTube one. Okay, Jessica Waters. I don't know. Okay, could we also include the use of artificial intelligence in the creation of synthetic vaccines as an application in the prevention paradigm? I mean, yes. I mean, there is a lot of work that is going on in the vaccine design as well, right? So yes, it is possible. To do this. I don't know much about AI applications in vaccine design, but I have seen quite a number of papers which do that. Yes, it is possible. Yeah.
Thank you, sir. There is a small
Yeah.
disturbance from my side.
Yeah, no
Okay, sir.
problem.
What is the difference between machine learning and you explained about what is the role of machine learning in drug discovery. Harshini is asking one more question, sir. What is the
Okay.
difference between artificial intelligence and machine learning? Both are same, or we consider both are same, or both are different? How it is helpful in drug discovery process? If you talking about artificial intelligence and machine learning, how both are going to help in drug design process or the drug development process?
Okay, so artificial intelligence is sorry, machine learning methods is a class of methods which come under artificial intelligence, right? So it's like artificial intelligence basically means that the study of how you make machines do the things that would normally require human cognition, right? So machine learning methods are uh, you know area within artificial intelligence where you use a large amount of data and then you try to learn from the data so that you can do the tasks, right? So if you take deep learning, that is again a small subset of machine learning methods are called deep learning methods. Yeah. If you take
Thank
the
you, sir.
artificial intelligence as the big picture, machine learning is a part of it, and then if you take deep learning, that's a part of machine learning. Yeah. So let me put it that way. Yeah.
There is one more question from Uma madam. What she is asking is, what kind of support do we need to develop a program for target identification and considerations required for how we have to identify a target based upon this machine learning process or AI?
Okay, so see, one of the things that one of my colleague does, I don't work in that area so much, but I do collaborate with him. He's Dr. Vinod from Triple IT. Maybe you can look up his webpage and look at his publications. So one of the early stages of target identification is to understand the biology of that particular disease, right? If you take, for example, the coronavirus 2, we try to understand how the replication cycle works, right? Starting from infection, which proteins are responsible for this host cell and the virus to stick together. From there, all the way to release of new virus particles from the host cells, right? So we study the complete cycle and then try to understand the pathway in terms of all the interactions between the viral and the host system, right? Okay, so and then we try to under this. You can think of this like a network of different entities, right? It could be proteins, RNA, it could be anything. Okay, so now in this network, you want to interfere with this network, and then a certain pathway within this network, such that it doesn't progress. Okay, so there also, to analyze, all the drug protein interactions that exist that we know, and all the other, interactomes within a host cell and then within the virus, and how these two two pathways interact with each other. There are, a lot more information that we cannot really, comprehend ourselves, right? So in that sense, you can combine a large number of these combine a large number of these information from these networks, drug networks, drug protein networks, protein networks, and then the viral human, interactome and so on. You can combine all these things and try to identify targets which, if you would intervene, will stop the disease progression. Right? Yeah. Again, there are some very nice papers that you can look at in this area.
Okay, sir. Thank you. One more question, sir. What are the institutions that are offering this artificial intelligence? Any institutions are there? Any specific courses are there which are offering this artificial intelligence? Separate courses. What are the organizations that are offering? If you have any idea, please let me know.
Okay, so institution means, I don't know if the context is a program by itself or short-term trainings. I do not know the context, but there's a large number of, almost all IITs and even IIIT Hyderabad, and a lot of us offer training in artificial intelligence and machine learning. Okay, we have these programs which we run once in 3 months, right? This goes on. This is like more for professionals, actually working professionals, right? And then we also we are planning to actually conduct a 1-week training school or winter school or summer school, in July. But due to this COVID-19, we could not do it. But I cannot think of a program that is meant for pharmacy students or meant for science people. But then in general machine learning programs and AI programs, a large number of them are available, both physical and online, right? Yeah.
Okay, sir. Thank
Including
you. One
one
more
from
question.
Hyderabad.
Yeah. Okay, sir. Thank you. There is one more question from Sai Supriya.
Yeah.
Will the drug discovery can be done in shortest time by using AI? That means by using this artificial intelligence, it is possible to reduce the time in the discovery process.
Yes, absolutely.
How AI will be helpful?
Yes, absolutely. Because what is the process in the early stages of drug discovery or drug design is basically that you want to identify one, or you want to identify few molecules that have strong affinity to that particular target, right? So basically what we are doing, in some sense, it is a search. We are among a large number of possibilities, we are doing a search. Okay, so there are a large number of these AI methods which allows us to make this search faster, right? And then the second answer to that question is my last topic that I showed, the inverse molecular design, right? So traditionally what we do is, if I have let's say 1 million molecules, right? And then I want to identify, let's say, top 1,000 that would interact with this particular target. Okay, so normally what we would do, let's say I do a docking, right? I take all these 1 million compounds and then I do docking on all of them, and then I rank them and choose the top 1,000. That's what we do, right? So if I take this particular method that I showed at the end, okay, maybe I need to do only docking for 20,000 or 10,000 or 30,000 compounds. Okay, so based on only that data, now I can go back to the original data set of 1 million compounds. I can do a search there and then pick the molecules which will bind strongest. That's one example, of course, but then there's the process itself in terms of finding the molecule which would bind strongly to a target, and also finding molecules with desirable pharmacokinetic properties. Okay, so it's possible to combine all these objectives and then make the search much more better and efficient. Okay.
Okay, sir. Thank you, sir. One more question is there from Indrajit Saha. What he is asking is, can MI predict receptor binding affinity of a protein molecule?
Receptor binding affinity of a protein molecule. I do not understand if
what
he
his idea is. I think he is asking that whether machine learning, by using this machine learning, is it possible to predict receptor binding affinity of a protein molecule?
Yes. So if you have a protein molecule and let's say it has the binding site, okay, So especially in case of small molecules, small drug-like molecules, it is possible, or there are some machine learning based scoring functions that are available which can quickly estimate the binding affinities, right? But again, as all said and done, this is an research area which is growing now, right? It's not like we have all the solutions, or artificial intelligence has provided all the solutions and we have everything, right? So this is an area where there is a lot of potential and a lot of work is being done, right? So we will I think, or most of us believe that this will make the process faster and much more efficient.
Okay, sir. Thank you. There is a question from our principal, madam. What is the type of machine learning used to know property of required compound?
Properties of required. Okay, so what I think what she means is, what are the algorithms that we can use to pick molecules with a certain property, right? So
Yes,
there are two three main methods that one can use. One is what is called variational autoencoders, right? And there are quite a few papers now on that subject, right? And the second one is GANs, GAN, right? GAN. And then the third one is what I explained today. That's what is called the reinforcement algorithm. And people also are using, and we also use what is called Bayesian optimization for this kind of problems, right? So again, as I said, we are slowly moving towards having more and more efficient methods. Yeah.
Okay, sir. One more question from Sharat Contra. What is his question is, what is the difference between CAD and drug discovery with respect to the artificial intelligence?
I mean, the computer-aided drug design so far. See, I mean, if we look at before 90s, right? We never used docking. Okay, so when we say computer-aided drug design, I don't really know if computer-aided drug design, that particular term, actually existed or not. But if to think of it today, maybe the QSAR methods, structure activity relationship was what was done before, right? If you take after 95s, especially if you take, we started using docking calculations, and then the 3D QSAR calculations, right? And then we also started predicting toxicity, right? We started having toxicity models for toxicity and so on, right? So if you look at today, we have one more tool, right, in this computer-aided drug design, which is machine learning based methods, right? That's what I used to say also, right? We only add more tools to our toolkit and our skill set, right? So if we have more methods that can solve a problem, right? So we don't drop the old methods. Of course they'll exist, they will always be useful, right? So how we use, when we use what, is going to determine. It's just that we are adding one more tools to our toolkit, right? Which is machine learning methods to computer-aided drug design. So that's how I look at it. Yeah.
Okay, sir. There is one more question from Manisha. What are AI neutral networks? How they are used in AI?
So neural networks, I did not go into the details, but neural networks, artificial neural networks, are the ones which, if you remember, in one of the slides I showed, if you have examples of X and Y, right? I said machine learning is the process by which figures out what that F is, right? The example that I gave was a set of numbers. Then I said, if you multiply X by 5, you approximately get Y, right? So that example I gave. So in that case, the function is multiplication with 5. But then the prediction tasks that we do in drug design, or in principle, other areas of science, is much more complex, right? What I mean by much more complex, the number of parameters are so huge, right? So one of the examples I showed, is like the molecular representation using some kind of like different dimensions. No, so in that case, what is my x? My x is like different kinds of bonds, different kinds of atoms, different kinds of angles, different kinds of non-bonds, right? And then the values that each of these things can take. So the number of variables is really large, right? So in principle, it is f of x1, x2, x3, x4 up to xn, n being a very large number. So now I'm predicting y based on this large number of values of x's. Okay, so you cannot, by looking at it, you cannot really say it, right? That f there is the neural network. That's the function. That's a complex function, or now like a universal approximation, right? You use that is your neural network. That simply is a collection of neurons. In this case, they accept some inputs and give some outputs, right? If you combine a large number of neurons, okay, so that again has large number of parameters by which it can take values of x1, x2, x3 up to xn and give you value of y. So all these neurons have adjustable parameters, and those are adjusted based on the difference between the actual y and the predicted y. That's how we try to come up with a function f which is suitable. Okay, so I don't want to give a much more mathematical explanation to this, but yeah, I hope you get the idea.
Thank you, sir. Thank you very much for answering all the questions. Thanks a lot. And we are very much thankful to you for accepting our invitation. You elaborated the topic. Thanks a lot, sir.
Oh, thank you very much for having me. Yeah, thank you very much. Yeah, okay.
So thank you so much, sir.
Yeah, thank you. Thanks for having me, Dr. Sumakant.
Yeah, your acceptance and talk, it's very clear, and I think many people know that nothing is there in machine learning. We can also do the machine learning. That is the conclusion after your talk, sir.
Oh yes, absolutely. Anybody should be able to do. It's not very complicated, as we
Pharmacist,
yeah.
I can say that it is very helpful for the students.
There is some disturbance, I think, right?
Yes, sir. Yes, sir. There is a disturbance.
Yeah.
Okay, sir. Thank you very much.
Yeah, welcome.
For your contribution to our webinar. Thanks a lot for accepting and your professional contribution to the webinar, sir. Definitely, we can always, whenever there is a possibility, we expect the same kind of cooperation from you wherever possible.
Sure. Yeah.
Thank you, sir.
Okay.
Thanks a lot.
Thank you. That's nice. Yeah. Bye.
Other participants, tomorrow also you please log in at 11 o'clock to our YouTube channel. Please continue the journey with us for this 1-week webinar session. Thanks a lot. With this, I would like to wind up today's webinar session.